Welcome and thank you for joining us for the last Agro Lunch and Learn webinar of 2022. This webinar series is only possible through the generous support from Eurofins Agroscience Services, and we thank Eurofins very much for their continuing sponsorship. My name is Solito Sumalong with AgRauthority's Regulatory and Product Development Services, and I'm an officer for the ISAA Agrochemical Network and an executive committee member for the ACS Agro Division. I'll be serving as moderator today for the webinar. Attendees should use the Q&A tool to submit their questions and comments, which will be answered at the end of the presentation. Before I introduce our speaker, I'll highlight benefits of becoming an Agro member. Agro pays enormous benefits by enhancing your science, building your scientific network, and supporting your professional development. Please go to agrodiv.org and click on Become a Member to become an affiliate for $20 a year, regardless of your membership status with the American Chemical Society. The ACS Spring National Meeting in Indianapolis, March 2023, is a rare opportunity to experience our agro-technical content during the spring meeting. On Wednesday, the 29th, Corteva is presenting an Agro Division social event with refreshments, fun, networking, and prizes at their campus just minutes away. If you have any questions about programming, contact Ken Rocky or Aaron Gross of Virginia Tech for agro programming question. For the fall meeting, we have the standard excellent agro programming, as well as the rescheduled 50th birthday party of the Agro Division. If you have any questions about the programming, contact Aaron Gross of Virginia Tech, who is the program chair for both the spring meeting in Indianapolis and the fall meeting in San Francisco. Now, I am proud to introduce our speaker. Sarah McCoy is a staff scientist at Eurofins Agroscience Services and coordinates their aquatics department programs. She has a master's degree in molecular biology and genetics from the University of Delaware, as well as research background in biomarker discovery with extensive experience in molecular biology assays, including RNA sequencing, qPCR, and protein quantification methods. Through her background and acquired experience in aquatic ecotoxicology, she is integral in delivering results for the multidisciplinary biologics testing program for Eurofins Agroscience Services. So, no further ado, I'll hand it over to Sarah.
Thank you, Solito, for that introduction. I'm going to quickly share my screen here. So thank you, everyone, for joining today. I appreciate your time and listening to this talk. Like Solito said, my name is Sarah McCoy, and I'm a staff scientist for Eurofins Agroscience Services at their site in Easton, Maryland. I do have a master's degree in molecular biology from the University of Delaware, and I have previously worked in academic settings in genetic research labs with experience in qPCR, RNA sequencing, cell culture, and protein quantification. I have been with Eurofins now for about 5 years. Here at Eurofins, we offer a wide range of ecotoxicology testing services, and at the Easton site, we specialize in aquatic, avian, and plant studies as well as terrestrial invertebrates. We pride ourselves on being able to offer comprehensive testing strategies that keep up to date with current guidelines. We have seen an increasing need for the testing of biological plant protection products. So today I'm going to talk a little bit about that and about some of the challenges we face when considering ecotoxicology testing of these types of products. So to start with a brief outline of what I'm going to be touching on today, I want to establish context and go over a bit of the background on what a biological plant protection product is, and a little bit more detail on which categories require testing for regulation under the EPA and OECD guidelines. I will touch on the numerous benefits of biologics that they can provide from a human health and environmental safety standpoint, as well as an important component of crop management and sustainable agriculture. I will also highlight the diversity of these types of products and some of the challenges that diversity and hurdles that diversity puts in our way for their widespread use. I will talk about the current regulatory framework in the United States, as well as considerations needed for an environmental safety testing. And finally, I'll highlight some of the gaps in our knowledge and present ideas for standardizing future testing. So what are biologics? Biologics cover a broad spectrum of agricultural products that are naturally derived. We can separate these into three main categories. A biostimulant is a term that's used to describe any natural substance that is beneficial to plants but does not provide nutrients or act as a pesticide. It is a product that acts on the plant itself to enhance nutrition efficiency. It can increase abiotic stress tolerance or improve crop quality traits. Currently, there's no harmonized regulatory framework that exists for biostimulants, and the criteria used to determine the need for registration is dependent on the product itself and its capabilities. However, efforts are underway to establish harmonized regulatory framework for these products. Biofertilizers are any biological product that enhances nutrient availability and promotes growth of the plant. They tend to be microbial in nature, and the active ingredient can be the whole organism or metabolites that are produced from the organism. Like biostimulants, there is currently no harmonized regulatory framework for biofertilizers at this time. For the purpose of this talk, I'm going to focus on biocontrol products or biopesticides. They require environmental safety testing and have regulatory requirements that are more closely aligned with what we're used to with conventional agrochemicals. So what constitutes a biopesticide? Biopesticides or biological pesticides are pesticides that are derived from natural materials. They can be animals, plants, bacteria, and certain types of minerals. They are categorized as pesticide based off of their method of action and if they have any pesticidal activity. Macroorganisms that are pesticides are not currently regulated as biopesticides, so we will focus on the three main categories that are microbial pesticides, biochemical pesticides, and plant-incorporated protectants. Microbial pesticides are microorganisms such as bacteria, fungi, or viruses that have a pesticidal effect. They can have modes of action that are competitive, inhibitive, toxic, or parasitic. Each type of microbial pesticide is relatively specific for its target pest. Biochemical pesticides typically have a non-toxic mode of action to the target pest and are generally considered safe for humans and the environment. They include, but are not limited to, semiochemicals, plant and insect regulators, repellents, and attractors. They typically degrade rapidly and are not persistent in their environment. They tend to have a broader spectrum of application when compared to other types of biopesticides, particularly microbials. Plant-incorporated protectants are substances with pesticidal activity that are produced by the plant or seed itself through bioengineering techniques. The active substance and the genetic material required to produce it are what are regulated as pesticides, not the modified plant or seed itself. So biopesticides provide many benefits over conventional chemicals. Managing pests in ways that do not adversely affect human health, beneficial organisms, or the environment is an increasingly challenging goal. Biological pest management products can offer solutions that minimize impact on non-target organisms and the environment, eliminate toxic residues, and are less prone to pest resistance. So we're going to touch on each one of these benefits in more detail. The specificity of biopesticides generally decreases the amount of product that is needed for use. It also eliminates the broad spectrum effects of a conventional chemical, which vastly reduces effects on non-target species and other beneficial organisms. The specificity can be driven by sequence, as is the case in RNA interference, where a double-stranded RNA product can impact gene expression in a target organism or pest by targeting sequence-specific mRNA for degradation. It can also be driven by environmental and protein-to-protein interactions, which is the case for Bt toxins. Bt toxins are produced by the bacteria Bacillus thuringiensis, which is a naturally occurring spore-forming microbe that has been used in a plant protective role for years. The pH of the gut environment of target insects allows for proteolytic cleavage of Bt proteins, which then allows them to bind to receptors on epithelial cells of the gut of the target insect. This binding allows them to form pores within the gut lining, which leads to osmotic cell lysis and eventual death. So different strains of Bt microbes produce proteins that are specific for different types of insects. The biological properties of the biopesticide itself, as well as the biological properties of the target organism, can be utilized to optimize the effectiveness and specificity of these products. Biopesticides are generally considered to be more environmentally safe. Biopesticides are considered more environmentally friendly for many reasons. They tend to be biodegradable and less persistent in the environment when compared to conventional chemicals. They pose little to no risk to birds, mammals, and beneficial insects, and they contribute to the decrease in the use of chemical products, either by replacement or as part of a pest management program. This can lead to a decrease in contamination of soil and water of both biological products and conventional chemicals. From a sustainability perspective, the production of biopesticide products is largely considered to be more environmentally friendly. An important role of biological pesticides is in residue management. The EPA sets tolerance limits for the amount of pesticide residue that can remain in or on treated food products. Exemptions from residue limits are granted when a product has been found to be safe. The EPA has established tolerance exemptions for registered microbial pesticides, genetically modified microbial pesticides, and plant-incorporated protectants because, based on thorough scientific evaluation, it has found that the tolerance exemptions are generally safe. Also, as a byproduct of the reduction of conventional chemical use, biopesticides indirectly decrease the chemical residues on food as well. Chemical resistance is an increasingly larger issue in agricultural pesticide use. So biopesticides can be very beneficial in resistance management. They can reduce the occurrence of pesticide resistance to conventional chemicals when they're used as part of an IPM program. In an IPM program, biopesticides are used in rotation with conventional chemicals and thereby decrease the amount of time target organisms are exposed to the chemical, as well as targeting pests that have developed resistance and therefore prevent resistance persistence in the offspring. Biopesticides themselves are more likely to have novel and complex modes of action and therefore are less likely to have problems with resistance in target organisms. Pesticide resistance can develop in multiple ways as illustrated in this graphic. You can have behavioral resistance, penetration resistance, where the product is no longer able to enter the target area as effectively or efficiently. You can target site insensitivity, where altered receptors or knockdown resistance. You can have symbiotic mediated detoxification, where a symbiotic organism mediates degradation of toxic compounds or immunity. Genetic resistance, a mechanism by which novel mutations mediate resistance to a toxin, or transcription factor mediated resistance, where you have overexpression of a particular set of proteins or protein in response to a toxin, such as in an immune response. This can also include epigenetic modifications that can control gene expression of response proteins. It's important to note here that resistance can develop via these methods to biological products as well. So having a diverse toolbox for pest management is crucial to avoid resistance development. We've talked about how biopesticides are safer to non-target organisms in the environment, and this holds true from an occupational safety perspective as well. There's a reduced need for personal protective equipment during application, which can be a cost savings. Biopesticides also have reduced restrictions on re-entry time after spraying. Most often, this is less than 4 hours. This provides greater flexibility for laborers, particularly with crops that require multiple harvest times, such as fruit. There is also little restriction to when pesticides can be sprayed before harvest because of their safety, so increasing the amount of time in which pesticides can be applied and can be particularly useful in targeting pests right before a harvest. Integrative crop management, ICM, is a holistic approach to crop management, and it does exactly as it says. It integrates agricultural management practices such as site selection and crop rotation with crop nutrition, soil and water management practices, as well as with energy and waste management practices, along with pest management practices. So IPM is an important role in this pest management practice. Biopesticides are an important component of these IPM systems because of their impact in reducing the use of chemical pesticides and their role in resistance management. Biopesticides help increase crop yield in a similar manner as conventional pesticides. It can manage pests as part of a crop protection strategy. However, they have the added benefit of doing so in a more environmentally friendly and sustainable way. They help meet the consumer demand for organic food products and improve food quality and safety through residue reduction. Some biopesticides promote plant health through the acquisition of new traits or improvement of beneficial traits. They help maintain beneficial insect populations through the reduction of use of chemical pesticides and their own decreased likelihood to have off-target effects. In these ways, biopesticides can contribute to more sustainable agricultural practices. Despite the many benefits to biopesticides, there still remain significant hurdles and widespread challenges for their adoption and use. One of these challenges is a competitive market. The agrochemical market is crowded and competitive. And although it is relatively cheaper to bring a biological pesticide product to market when compared to conventional chemicals, it is still costly, and navigating the regulatory requirements can be confusing. Historically, biopesticides have been developed and produced by smaller to mid-level companies that have less resources and have more difficulty gaining attention by distributors for single or few products. The specificity of biopesticides, which is beneficial from a safety and sustainability perspective, can limit their use in comparison to broad-spectrum pesticides and therefore diminish their market value. The perception that the cost-benefit ratio is too high when it comes to biopesticides is still a consideration. To address some of these concerns and to promote the use of biopesticides, some countries have simplified the requirements for registration of biopesticides. Some countries also provide grants for R&D and production of biopesticides and setups. And some are also implementing efforts to reduce the use of chemical pesticides, which increases the usage of biopesticides, which may drive the market and create opportunities to develop the biopesticide market globally in the future. Another challenge is the application and use of biopesticides. Biopesticides are most effective as part of an integrative pest management system, but implementing these systems can require a great amount of skill and knowledge. Timing, accurate assessment of pest species, and assessing environmental conditions are all key components of using biopesticides effectively and can be a barrier to proper use. Overall perception still remains a challenge as well. Growers have years of experience with conventional chemicals and are knowledgeable in their use and application and effectiveness. It may require a mind shift change to consider biologics as effective as their counterparts and to include them in pest control programs. Assessing effectiveness of biopesticides by directly comparing them to conventional chemicals can cause a misperception. For example, some biopesticides take longer to cause a toxic effect or may be more effective if used as a preventative or at a specific time of crop growth or pest life cycle. Other situations may require multiple biopesticide products with different target pests and or modes of action, where a single conventional chemical might have been used previously. Thought and consideration is needed to consider the cost benefit of multiple applications at a lower density. According to this survey conducted in the Farm Journal approximately a year ago, 41% of farmers that responded specified that they still needed to know more about biologicals before they would be willing to consider using them. Educating users on the benefits of biologicals and their proper methods of application will begin to change this perception. Despite these challenges and the fact that the biopesticide market makes up a smaller percentage of the agrochemical market, the biopesticide market is expected to more than double in the next 5 years, as shown by the $6 billion increase between now and 2027. This growth will be driven by consumer awareness, increasing issues with pest resistance, and the impacts conventional chemicals have on human and environmental health. The need for ecotoxicology and environmental safety testing will grow with this demand, which presents an opportunity to evaluate the current challenges we have with regulations of biological crop protection products and to discuss possible solutions for the future. Before a biopesticide can be marketed and used in the U.S., the Federal Insecticide, Fungicide, and Rodenticide Act, FIFRA, requires that the EPA evaluate the proposed pesticide to assure its use will not pose unreasonable risks of harm to human health and the environment. This is including non-target species. Microbial, biochemical, and plant-incorporated protectants are considered pesticides under FIFRA and generally are required to be evaluated and registered by the EPA. Currently, the United States EPA is the only regulatory body that has separate guidelines specifically for the registration of biologics. Current guidelines allow for flexibility in fulfilling data requirements. This can be accomplished by providing a scientific rationale why tests are impractical to perform or to address a particular endpoint. In addition, additional testing requirements will be requested if potential risks need to be investigated. These guidelines were established as a framework purposefully. The diversity of biopesticides provides a testing challenge, and a comprehensive approach is needed when undergoing toxicology testing to ensure that unique characteristics of the test material and organism is considered. Ongoing efforts are being made to harmonize the regulation of biopesticides. The EPA and Health Canada's Pest Management Regulatory Agency, PMRA, have established a process for the joint review of biopesticide products. Joint pre-submission meetings are encouraged to establish data requirements. The US EPA and EU OECD also participate in a biopesticide steering group, which meets annually to discuss harmonization of guidelines and principles of risk assessment. This schematic is a rudimentary attempt to acknowledge that although I'm going to discuss that we need more prescriptive guidance and harmonization among regulatory bodies, there is ongoing work in this direction by many groups. For simplicity and the purpose of this discussion, I'm going to narrow our focus at this time to the regulation of microbial pesticides specifically by the US EPA. So just to further define, a microbial pesticide is a subset of biological pesticide that can be a bacteria, fungus, virus, or protozoan. The unique nature of microbial pesticides has led to changes in data requirements. Regulatory authorities generally require sufficient information on a microbial pesticide to characterize it, to assess its potential risks to people and the environment, and to confirm its effectiveness for pest control. Unlike chemical pesticides, microbial agents may infect or cause disease in other living organisms. Potential adverse effects of these microbial pesticides include displacement of non-target microorganisms, as well as toxic and pathogenic effects on humans and other non-target organisms. Consequently, the risk assessment of microbial products rests heavily on the biological and ecological profile of the microorganism and a set of short-term toxicity and pathogenicity tests, with the expectation that negative test results will allow for a high degree of confidence in the safety of the microorganism. Like conventional pesticides, specific criteria is given for what type of data is required for submission. The flexibility in the guidelines allow for waivers to be given based on available scientific data or requests for additional data if deemed necessary. Identification. For microbial pesticides, correct identification is crucial. The most accurate taxonomic information is needed to verify the identity of the active microbial agent at the strain level. Information on genetic modifications is necessary, as well as traits that have been acquired due to that modification. Any adverse effects known to be associated with a microbe or closely related species is required. Information on manufacturing, discussion on formation of impurities is also included. For microbials, this information should include an examination of methods used to verify the purity and the stability of stock cultures. This is to ensure that contamination is avoided. Genetically modified organisms may be subject to different data requirements, such as information on techniques used, identity of inserted and deleted gene segments, expression control regions, new traits, genetic stability, and possibilities for environmental expression. Pre-submission meetings can be important to discuss and clarify data requirements. This complexity illustrates the importance of having an understanding of the biology behind the test product and to determine what types of testing may be necessary. The data requirements for microbial pesticides are tiered, where Tier 1 tests must be performed and higher tier testing is only required if adverse effects are seen at lower tiers. Tier 1 data requirements include tests on birds, aquatic invertebrates, fish, non-target plants, and terrestrial insects, including bees. Rarely have biopesticides been subjected to testing above Tier 1. Other non-target organisms, plants, and beneficial insect testing are considered or required depending on the proposed use of the product. These tests are described in the U.S. Environmental Protection Agency's Series 885 guidelines for microbial pesticides. Because microbial pesticides are derived from a range of naturally occurring microorganisms that include bacteria, fungi, viruses, and protozoas, the guidelines to conduct these tests are not overly prescriptive, but try to provide latitude to adapt tests based on the nature and hazards of each active ingredient. The regulatory framework around microbial pesticides and the 885 guidelines are intended to be flexible to allow for considerations of the diversity of the products being tested. However, that flexibility can be challenging from a practical standpoint when conducting ecotoxicology tests. The framework is valuable to make a comprehensive assessment, but there is a need for more specific criteria in conducting these tests so that they are standardized between facilities for similar types of products. To illustrate these challenges, I'm going to use a loose comparison of the Aquatic Ecological Effect Testing Guideline, OCSPP 850, and the Microbial Testing Guideline, OCSPP 885. As an example, first of all, we know what we're doing at CROs and in regulatory agencies when we're dealing with the 850 guidelines. We have years of experience working with conventional chemicals, and these guidelines have been updated over time. Also, the guideline is very prescriptive and provides clear criteria for conducting these tests. It includes information for how to conduct range-finding studies and to establish test concentrations, including criteria for what constitutes a successful range-finding study. Selection of a specific test or organism. The organisms in these tests are familiar, have historical control data available, and there's specific criteria listed for acceptable health and robustness of organisms before use. The guidelines specify that preliminary work should be done to determine the behavior of the test substance under test conditions and give specific criteria for the preparation of test solutions, as well as guidance on what to do if the material is insoluble or test concentrations cannot be maintained. It lists specific criteria for acceptable analytical methods, method validation, and acceptable criteria for analytical recoveries. The endpoints listed are mortality, growth, and reproduction, and the definitions are listed for how to assess each endpoint. Clear criteria is also required for acceptable controls. The microbial testing guidelines states that species selected should be closely related to the target, should be as closely related to the target host as possible, and that some of these species likely to prey on the disease target host should also be considered for testing. However, this is an issue because the guideline gives no guidance on how to determine what species would be best. How do we discover closely related organisms, and how closely related is acceptable? Also, we need to consider the challenges of testing new organisms and consider that the same organism may not be selected by different testing sites. The guideline specifies that the test substance should be administered as a suspension in water. This is an issue with a lot of microbial products that exist as spores or may not be soluble in water. It will be hard to maintain a suspension for testing. The maximum hazard dose is defined as 10 to the 6 units per mil, or 1,000 times the maximum calculated concentration in 6 inches of water immediately after application to water, and that the initial concentration of that product should be maintained throughout the test. When considering microbial products, it may be difficult to maintain test concentrations because there's a possibility of the test substance to come out of solution. It may replicate in the test system, or it may die and degrade rapidly. The test duration is extended to account for the time needed for potential pathogenic effects. However, this can be an issue for some target organisms, specifically terrestrial bees. No clear criteria is given on analytical methods or the acceptable recoveries. For example, the conventional method for microbial quantification is plating and colony forming unit counts. This method can be highly variable and does not account for contamination. There may be a need to apply multiple different methods of quantification or institute new technology and different types of quantification methods. Clear endpoints are given in these guidelines, but no guidance on how to determine what infectivity or pathogenicity may look like in different organisms. This flexibility can lead to inconsistencies in testing methods between and within CROs and can diminish the reliability of the data. So to reiterate the specific challenges that we see, solubility is of particular concern when conducting aquatic tests, but considering the delivery of the microbial can be applied to all types of testing. How the microbe interacts with a test system will have implications on being able to properly maintain a test concentration in the system. Utilizing closely related non-target organisms for testing is extremely important considering the specificity of microbial pesticides. However, specific criteria for deciding on which organism will be helpful. Implementing the use of standard tools and technology to do this assessment will also be valuable. The guideline specifies that the active ingredient should be tested. However, considerations must be given for testing formulations of microbial pesticides. Sometimes the formulation enhances the delivery of the product or is required for the organism access to the target pest. It can even mediate its toxicity. The test system needs to be considered when deciding on what types of controls to use, whether a filtrate or an attenuated heat-killed organism would be more useful. We must consider the environmental conditions of the tests, as they may be suitable for the test organism, but not for the test product itself. Analytical criteria needs to be more prescriptive, and new methods or combinations of methods should be utilized. The endpoint should be evaluated based on the expected mode of action of the microbial pesticide. Is it meant to be generally toxic or cause a pathogenic reaction? Contamination during testing and analysis should be of concern. It can affect the activity of the microbe, compete within the test system, and impact analytical confirmation. So moving forward, it's going to be really important to establish tools to select closely related organisms that are being considered for testing. We need to consider holding, food, temperature, and information on how the target species and how closely related they are. More prescriptive guidance is needed. Trials before testing how the microbe reacts in the test medium. We need more prescriptive criteria for method validation. We need to utilize available technology, which can include bioinformatics, modeling, and current databases to predict outcomes. As a CRO and research scientist, we can assemble multidisciplinary teams, acquire different testing capabilities, partner up to conduct proof of principle studies. So if we're considering a certain analytical method to determine how it compares to CFU counts, establish methods for organism selection and alternate organism holding, rethink our analytical methods, and actively consider the biology of a product, an intended method of action, and unintended consequences. The test guidelines provide an important framework in which specific considerations and challenges are taken into consideration, and still allowing for flexibility in testing. However, more prescriptive guidance on the steps needed to take or a decision-making pipeline would be useful. And thank you so much for joining, and I'd be willing to take any and all questions at this time.
Thanks, Sarah. We did have a few questions rolling in, and I had a few as well moving forward. I think this is a very fantastic overview of the regulatory and data requirements to register a biological in the U.S. market. Some vendors are now using the combination of chemical pesticides and biologicals. Which type of specific regulatory guidelines do you recommend for these combo products?
Well, the guidelines actually, or there has been some discussion on what you do in that situation, and currently each active product needs to be tested separately. However, testing it as a combination, especially if there's a synergistic effect, might be useful as well.
I'll allow the anonymous attendee from the 1133 question to elaborate on this, but could you, Sarah, elaborate on the differences for toxicity, eco, animal, and human data requirement for biopesticides registration compared with the conventional chemical pesticides?
I don't think that I can give you a comprehensive overview of that, especially with the requirements for the human. But really the unifying theme of data requirements is that it's dependent on the test product itself. So they really encourage pre-submission meetings to kind of determine, what do we need to look at and what's going to be important for submission.
I'm going to combine one from Bettina Moser with the anonymous attendee here. Thank you for the presentation. Pertaining to EPA microbial pesticide test guidelines, I am curious how to differentiate symptoms of toxicity from symptoms of pathogenicity. I'm sure that depends on the test. And as a corollary to that question, which parameters do you look for potential early screens for evaluation of infectivity or pathogenicity?
So that's an excellent question, and that's basically one of the huge issues with these test guidelines is that there's no prescriptive, description of what that looks like. So depending on the organism you're testing, it may look different. We have, up to this date, we ourselves have never seen infectivity and pathogenicity with these test compounds, but that doesn't mean that it's not going to happen at some point. So, to look for pathogenicity, typically you would do, a histological analysis and look to see if the pathogen is, in the organism, and then other than looking for typical signs of infection, there's no clear criteria of what that's going to look like.
Yeah, that gets you to a question that I had from a CRO perspective. Let's make it personal. You're sitting doing your day-to-day with the aquatics program. And at the end of the day, particularly for a startup, right, Sarah, this is going to be an expensive endeavor to pursue this kind of data. What are your regular tests for clients, and where are they coming? What questions, particularly from the integrative pest management aspect, are they requesting testing for? That makes sense.
So if I can understand, so each product needs to be tested separately, even if they're being used together in an integrative program. Right. So we don't necessarily get the information on how the products are going to be used, other than, it's a pesticide or some information on method of action. So I think it would be important to know the method of action from a biological standpoint to determine what type of testing may be needed. And then to go back to your question, I'm sorry, what was the first part?
We were talking about what kinds of tests have been usually asked for with those limited resources.
Okay, so the tier 1 tests in the Microbial 885 guideline. So it's a short-term, approximately 20 to 30 day test with Daphnia, with some fish, and you're supposed to pick your organism based off of, a closely related target pest, but we typically do these tests with fathead minnows. And I'm blanking on terrestrial insects, which would be bees, just terrestrial invertebrates, aquatic invertebrates, fish. I think that's it.
Yeah, from Greg Watson, and this is also another curious aspect of registering biologicals, not only in the US, but globally. But are you aware of the recent OECD Cooperative Research Program funded workshop on innovating microbial pesticide testing? The consensus document and follow-up actions from this workshop are going to be important in addressing some of the issues you raised in the presentation. How much guidance do you give to your clients on the applicability of testing for multiple geographies?
So we try to give as much guidance as possible. And going forward, this might be more of a consideration with these types of tests where, with conventional chemicals, it's it's clear what needs to be done. You're following a set path. Whereas with these biologicals, it might be more of a decision-making process between regulatory agencies, CROs, and the client themselves of what do we think we need to present to demonstrate that this is a safe and effective product to use. And I am aware of the new documentation coming out of the OECD and would be very interested to see, what impact that's going to have going forward. And that's kind of to my point where I was saying, we are completely aware of, as a CRO, of the ongoing work and efforts that are being made by the regulatory organizations.
Sir, we had an anonymous attendee ask about MOAs for biopesticides or biologicals. It's not currently listed as a requirement. Do you see that changing in the future?
I'm guessing MOA is a method of action. Is that
mode of action?
Mode of action. I think that is going to be important for sure, because if something is meant to be toxic, like it produces a toxin or the organism itself is toxic, you're going to be looking for different endpoints than if you're looking for an infection or a pathogenic endpoint result.
Are biologicals tested for efficacy the same way as conventionals? How much do you get into the efficacy realm?
So we do have, Eurofins does have an efficacy testing lab. I'm not as familiar with that part of this process. I do know, I'm trying to remember, am I reading correctly, and don't quote me on this, but I don't believe the EU requires efficacy for biologic products, but the US does, but it comes into play in the tier 2 studies.
From your laboratory and over in Eurofins, do you have any insight on whether biologicals have failed in tank mixes with herbicides and other products that have a lower high pH value? Have you tested? I know you don't test from the Ecotox standpoint, but from the BizChem standpoint, have you been receiving requests for compatibility?
I do not know the answer to that question, but I can ask that question to the appropriate people, yes, and pass it on if someone wants to.
Are you aware, Sarah, and I wish I had my NPeers up as well for if you've seen a registration for double-stranded RNA products yet?
Yes, we are aware that they're out there, and that there have been requests for testing of those types of products. Yes.
Now, one thing we haven't gone over necessarily is whether the plant incorporated pesticide general approach. How do you determine dosing, and therefore how do you reach those endpoints from the testing standpoint?
So that's an excellent question, and one that actually I haven't really thought about. So plant incorporated protectants can be pesticides, and they may not be pesticides. So it depends on the trait that is acquired by the plant, whether it has a pesticidal mode of action. If it does, the regulatory body considers it a pesticide, and safety testing is going to need to be done. I really haven't thought about how do you need to then produce a pure, stock of that type of compound, even though it's coming from the plant, and what the dosing would be? I don't know. That's a great question. I'd like to look further into that.
Here's an interesting question. This one can go in any number of directions here. Thank you for the presentation. Which and how many metabolites are produced by a single microorganism to be analyzed for registration? I'm sure that those effects are taken into consideration at the testing once we get into testing.
Yes, so I can tell you for conventional chemicals, we look for, we do metabolite analysis and look to see, where the toxicity is coming from. And I would venture to guess that you would take a similar approach for, microbials themselves. As far as how many metabolites, I think it would depend on the way the microbe reacts in the test system and how that happens.
I'll shift a little bit of gears here. Are there any differences for test requirements for modified proteins or toxins versus the natural sequences? What's been your experience with those?
Yes, there can be additional requirements for the genetically modified materials, and those are listed. You just need to give additional information about, the sequence, what has changed, what the new traits are. But as far as testing themselves, there is an additional endpoint for, gene transfer and genetic stability, but that also comes into play in the higher tier testing. So if the product makes it to that level, it's considered.
Anonymous attendee rather early asked about how they can determine what studies would be needed for your active ingredient in preparation for a pre-submission meeting. From my perspective, you go into that pre-submission meeting with what you have and make your best arguments based on your prior and public knowledge of the species and go from there. There are a lot of levers that you can pull to advocate for data waiver arguments. But
yes,
did you want to add to that?
No, that's an excellent answer to that question, Salido. Thank you. Yes, I think you would go in with the information that you have and then determine what you need between what's not in your possession and what's not publicly available as information. That would be what would need to fill the gaps in that safety evaluation.
And typically a good CRO or consultant will guide you as to what limitations you have for the label based for the crop or the target pests or crops based on the data that you have. So there's 3 left on the board, so I'm going to try to wipe out 1 and 3 here. So the requirements for double strain biopesticides are still expected to be tested under the same 885 US EPA testing guidelines. And then of course, you mentioned in most cases, the testing doesn't go beyond tier 1. Have you dealt with any cases where it's gone beyond that, not only for double stranded RNA, but for others?
To answer the second part, no. And the first part, that's actually kind of a tricky question because I have read both, that depending on the type of product or where the double-stranded RNA is coming from, it might be considered a biochemical versus a microbial. So those are slightly different. There are 2 different guidelines. The 880s are for the biochemical pesticides. So if you have a microbe that is expressing the product that ends up being a double-stranded RNA and has, the RNAi mode of action, or if you're applying a double-stranded RNA, in its pure form to the system, they might be considered 2 different types of products. I hope that clarifies a little bit.
And I guess the last question's pretty important because you're in there every day working the projects that you have over at Eurofins. I guess is an opportunity to discuss about any interesting or unexpected effects you've seen in microbial testing, and or any sob stories or any cautionary tales that you might provide potential clients as we get into this realm of biological testing.
I would say one of the most unexpected results is that the formulation sometimes causes toxicity when the active ingredient does not. And depending on the organism that's being tested, that can be because of water quality or something else that's affecting the organism itself. But as far as infectivity or pathogenicity, we haven't seen anything crazy, but that's not to say that it's not coming.
Yeah, we did have a webinar earlier in this year about co-formulants in biologicals as well. So there is going to be a market segment, in my opinion, for those that's going to be ongoing, and I think the particular challenge in that case, and you bring that one up, there is that particularly for the co-formulants is the safety of the microbes in the formulation. That's certainly an additive challenge, not just the efficacy aspects, not just the compatibility aspect, but just keeping these things the microbes viable. So
yes, exactly.
Well, thank you so much.
Thank you for having me, and thank you everyone for participating today.
Let's close out the webinar, this webinar and the webinar series for this year. Thank you very much for your participation. I would like to thank Sarah McCoy for an excellent job today. Eurofins AgriScience Services, our webinar coordinator Kyle Sawyer, and the webinar committee for the entire 2022 program. You may be asked to complete a follow-up survey after the webinar, and we look forward to your feedback on this webinar and all of our 2022 webinars. This happens to be my first year being the chair of this series, and I'm so humbled to have been in a position to keep the ball rolling. 2023 webinar planning is going on right now, and we do plan to present 6 times next year. You see the dates tentatively. Kyle has confirmed those dates with me, so we will operate with those dates for this year. We do plan on presenting on overlapping insecticide, herbicide molecular targets from Stephen Duke. He will be our March 8th presenter. And also we have webinars on biologicals as a continuation of Eurofins' series on biologicals, as well as ISA 8 Agrochemical Networks co-sponsored webinar on agrochemical formulations and inerts. Thank you all for your attention today. Thank you for all for your attention this year, and we will see you all in 2023. Thank you very much for joining us, and have a happy new year.